Good morning, everybody. I'm Lori Burrows. I'm the Associate Director of the IIDR, and welcome to the April edition of IIDR Rounds. So our speakers today are Dr. Des Pernica, who's a pediatric infectious disease specialist and an associate professor in the departments of Pediatrics and Health Research Methods, Evidence, and Impact. He is also a member of the IIDR, and he's going to be talking to us today about discussing vaccine hesitancy with families. Very timely. Our second speaker today is Dr. Sam Afkhami. He is the operations manager for the Global Nexus at McMaster and is a senior research scientist at the Canadian Pandemic Preparedness Hub. He got his PhD with Zhou Zing, also our IIDR member, where he focused on development and evaluation of respiratory vaccine candidates for tuberculosis. He's now working on McMaster's next generation inhaled COVID vaccine, which he'll be discussing with us today. So thank you both for joining us today, and I will turn it over to Des.
Thank you. Perfect. These are my disclosures, and these are my objectives. So this nifty paper came out, which said that, like, hey, vaccines have saved a lot of lives, 150 million in the past 50 years, which I'm sure is no news to many people here. If anybody wants, an update from, industrialized countries only, this was a paper published a couple of years ago looking at the United States, and it unsurprisingly found that, routine vaccines dropped all of these important conditions by like 90 to 100%. But as we have all grown to learn over the past 5 years, vaccines only work when they are actually in people. Now, the last time I gave a talk like this, I threw up these headlines, which at the time was talking about the biggest measles outbreak that North America had seen in 25 years. This one also had a good story. Now, of course, all right, we know that we are having much more problems with measles now than we were in 2019. Oh, look, and the animations are all screwy. So let's look at factors affecting kids getting vaccinated or not. Long, long ago, before COVID, okay, these reports came out talking about what immunization coverage was like in Ontario. And if you look at the ones that are administered in school, these three, uptake is not great. Like HPV was at 60% pre-COVID. If you look at the other vaccines that were administered mostly by primary care, we also weren't doing amazing. The red line here is where we would like to be. The green bars are where we were. And so some public health units are doing pretty good, but some were doing very bad. And so you see MMR here is not great, or at least the MM part, right? Because you need two of those to be up to date. By the time they got to be 17, a lot of those had caught up with that dose that they had missed, but then they were missing the 14 to 16 Adacel, right? And that was before this. I think we all remember how it went at the beginning of COVID, and it is not surprising that coverage got very bad, especially in school when nobody was going to school. So you can see here that all the vaccines that were delivered in school dropped off a cliff. Here, there were ones that got started in 2019-20 that never got finished, and 2020-2021 never got started. If you look at what happens with vaccines that are typically given by primary care, like in the family doctor's office, those also got a lot worse because it was really hard to see one's family doctor, right? And so you can see here MMR going down to the 50% range. All of this to say, okay, there are lots of reasons for children and anyone not getting vaccinated that don't have to do with hesitancy per se, right? And all of this is about access to care, right? We have heard about the millions of Ontarians who don't have a family doctor. If they don't, they're not being told when to come in for vaccinations. Um, Often people may have a doctor, but they don't have transport, they don't have coverage, or they don't have good rapport with the person who's giving them vaccines. And so they're not incentivized to go back. this is a major issue. People forgetting is not an education issue. I am, supposedly a doctor. I care about vaccines, and yet I forgot numerous times about vaccinating my own children because I'm disorganized. There is this is interesting thing of needle phobia in adolescence. We get referrals about this. So families who want vaccines, adolescents who apparently want vaccines but can't deal with the idea of needles. And this is not surprising. I'm not sure how big this is at a population level. This has not been assessed systematically to my knowledge, but it certainly happens and it interferes with them getting a vaccine. There are medical contraindications to vaccinations. The true contraindications are much are many fewer than the perceived. So I get lots of referrals, for example, for excessive crying after vaccinations. I mean, that is definitely bothersome to parents, but not really a contraindication. And I don't do much when I see them in clinic, except to say, like, I really think the benefit outweighs the harm of further vaccinations. The measles outbreak has actually brought a couple of these to the fore. So I've been getting calls, by a family who had never vaccinated their child against MMR because they have so many food allergies. Well, egg allergy ceased to be a contraindication to MMR vaccine decades ago. I get called by doctors who have patients with, seizures or autoimmune disease who hadn't vaccinated their patients previously because, those things could flare. Autoimmune disease can absolutely flare post-vaccination. Somebody with a seizure disorder can absolutely get a fever post-vaccination, which can provoke seizure. But again, these are not true contraindications to vaccinations. And so almost invariably, the potential benefit of vaccinating outweighs the potential harm. And then we get to this. We get to actual vaccine hesitancy, which is like intentional delay or refusal. And I think really there is a lot to think about here because vaccine hesitancy is so unbelievably complex and interesting. I think many of you know Ellen Amster, who's on faculty here at McMaster, who gave a much more interesting presentation on vaccine hesitancy than this one is. In the course of hearing her speak, I learned a lot about how there is really an extensive history of vaccine hesitancy, which is unsurprising when you think about how things started, right? Like they used to take a big, knife and scrape a cow and then scrape a person with it. They couldn't scrape a person and scrape a person with it because then you give them syphilis, which people don't like, right? And so that introduced a lot of these fears about putting this unnatural thing into a pristine child's body, right? That idea still persists today and is commonly invoked in contemporary discussions about vaccinations, especially new vaccinations, and this cartoon from, 200 plus years ago shows the same thing about how cows are erupting out of people because you're putting cow in them. Mandates, right? We think that mandate is a new thing. The first mandate for vaccination went in the 1850s in the UK, and the penalties were harsher than they were recently, which almost immediately provoked a backlash, right? In Toronto, in the UK, and this idea that the government is going to make you do something that's bad for you, that's bad for your family, obviously, okay, I provoke strong feelings in everyone. I mean, after the mandate was lifted for the COVID vaccine, I got referrals for years from parents worried that the government was going to put the mandates back in and their kid wouldn't be allowed to go to like university residence or something. And then this idea that who's benefiting, who's making money is a contemporary idea and an ancient idea. So before it was the doctors making the money. Now I think a lot of people realize that doctors don't do so hot, at least pediatric infectious disease doctors. And so it's big pharma. Why is big pharma doing all this stuff? Now, so all of that has been simmering for literally centuries, right? The internet, as I'm sure we are all aware, enables, much more dissemination of ideas, and especially by those who are media savvy, who know how to use television, movies, who have, big online followings, and it makes it easier for them to convert others. And then I just keep saying, and then social media made it worse. Like I think we are all aware about how the structure of social media has poisoned a lot of what we took for granted in our society today. I mean, aside from the fact that obviously nobody's fact checking anything. I mean, there was a little bit of fact checking for like a second, and now that's gone. But the structure, I mean, we all know this, right? Like I'm not a social scientist. Um, I mean, I'm not that smart. Um, the like inflammatory posts are much more likely to be either liked or retweeted, right? Which makes things, and then you get the anti-vax inflammatory posts, which themselves are also popular, but just doesn't help. Um, you just get the creation of these communities, and it's interesting. I've had these experiences where you don't see somebody for a few months, and then you have a chat with them, and it's shocking that the way that the discourse goes, and you wonder, like Aziz Ansari said, like, what algorithm are they on? And so this is the way that I feel. This is the way that I think that I feel, right? And yet, even though I am supposedly Canadian, I never really understand this whole thing. The medium is the message. I do now, right? The medium of X has totally shaped discourse. And then and then all of this stuff happened and magnified everything because the financial stresses got worse. People were dying all over the place. We had these brand new vaccines with brand new mechanisms of actions, which weren't really brand new, but were brand new enough to make people nervous. And governments, paying big pharma for all these things. And so it just felt like this. This GIF was the best thing that happened to me over COVID because it again encapsulates perfectly the way that I felt. And I'm a doctor. Like it was hard for doctors to figure out what was going on and what was true and what was not. And I was holed up in my apartment with my two kids, and I was writing these guidelines that changed every day. It was crazy. And so, in terms of conversations with actual families, these are some things that I have observed over the past while. Many of you may have seen this. This is an interview of the parents of the child who died in Texas, or one of the children who died from measles. And they are expressing how they still don't believe in vaccines, and the hospital may have not treated their kid properly. I guess what I would say is that there are some people that are in the pre-contemplative stage that are not in any headspace to have anything resembling a rational discussion about vaccines. And so, at this point, you can't really go that far. These people would never come to see me in clinic because they wouldn't care. Like there would be nothing for them to discuss. For the ones who actually are willing to talk, and there are a lot of these, right? I mean, I think the starting place is listening, right? I always ask to find out, to see if there's sort of a rationale that can start a discussion. Often they will have references. People used to have books. People never have books anymore, but it makes it easier to find the website because you can't leave the website at home. And these things I would always be prepared to confront, right? Post hoc fallacy is like the most human thing in the world, right? By that I mean, you're playing hockey, you don't shave, you win a playoff game, so you don't shave all that time after that because it must have been the beard that won you the hockey game. This is something that's been hardwired into us by evolution, so that we didn't get killed on the savannah or whatnot. I mean, the counter arguments to this are obvious. All right. Martha put me on to this website, this spurious correlations, which is always good for some laughs. You know, uh, you can find things for even like these functions that are multiphasic, and uh, often you can get people around, but there are sometimes where you just can't. I had a mother, like a totally normal parent, okay, whose kid somehow had come down with pneumonia after the influenza vaccine twice. Twice. I mean, it could happen, right? Um, and I just couldn't. I I just couldn't, even though it's obviously physiologically impossible for one to cause the other. You just can't get anywhere. Sometimes, there are some. The only good thing about immunizing, millions of people at once is that you have a good idea of what happens after they get immunized. And so sometimes parents are in the frame of mind, especially seizures post-COVID vaccine, because that happened a lot. Because seizures happen a lot. Um, I could pull up articles that would say that looked at, for example, the incidence of certain adverse events within a risk window, say 3 weeks, and compare that to the incidence of adverse events that happen outside of the window, say 3 to 6 weeks. And so for seizures, for example, there's no difference. And so it gives you a logical basis to say that probably the vaccine didn't cause the seizure. But sometimes there isn't the data you're looking for the vaccine that you're interested in. The inability to understand probability is a uniquely, well, not uniquely, is a very human phenomenon, right? This, again, is not an education kind of thing. Whenever I play poker, which is not that much, I always sort of think that I'm going to get a royal flush, which is obviously insane, right? There's no way that's going to happen. Poker players understand probability. The rest of us do not. And so for like Guillain-Barré and influenza vaccine, it is true that influenza vaccine can cause Guillain-Barré. Like that's happened in Hamilton, right? Unequivocal. Um, and you say to people, well, it's only 1 in a million. That just sounds like there's a small chance that something very bad will go wrong after you get the vaccine. Um, and when you can say, well, there's lots of people who get hospitalized and die from influenza, but a lot of people don't know anybody who's died from influenza. So when you tell that to people, they're like, well, there's this small probability that something bad's going to happen if I get the vaccine, but there's a small probability that something bad will happen. Or sorry, if I don't get the vaccine, but there's a small probability that something bad will happen if I do get the vaccine. And those sound about the same, even though the probability of something bad happening with the vaccine is much, much lower. People don't understand this. Sometimes you can say, well, there's this great study that I think Merrick was a part of that showed that you get way more Guillain-Barré from actual influenza, but you don't have this for every AFI, for every vaccine, right? And so people are like, well, I can't really tell which is worse because humans are like that. And that leads right into the next problem, which is errors of commission being hard. This is the trolley problem. I'm sure most of you know it. For those of you who don't, the scenario is that this trolley is going to come and is going to kill these 5 people who are tied down to the track. But this person here can flip the switch, and if the person flips the switch, the trolley will only kill this one person. But the person with the switch has a sad face because they don't want to flip that switch because if they do, it'll be their fault they killed the one person. Whereas it's not their fault that those 5 people die. Um, and this seems crazy, but it's very intuitive to me. And what I will say is that this often happens when you have one parent who's pro-vaccine and the other one who's not. And the one parent who doesn't want to give the vaccine is like, how are you going to live with yourself if something terrible happens? And so people delay because that's the easiest choice. The problem is that delaying past infancy robs the the people who need the vaccine most from the protection that the vaccine gives, right? And so this is very bad. And now we have this problem, which seems to be worse. Like I feel like more people think that I am Nick Riviera than did previously. It's harder for people in primary care because all I do all day long is talk about vaccines. So it's easier for me to know stuff. But like, family doctors have to know all of the things. So it's hard for them to have all of the answers, right? So you have these people who think that you're stupid for bad reasons. There's people who don't trust the healthcare system for very good reasons, because racism kills people in the hospital. And then you have the issue of doctors. Some doctors who think that school should have been closed for 3 years because COVID might do something bad to a kid versus the doctor who thinks that the COVID vaccine is going to give his kids myocarditis, thrombosis, cancer, and sterility. Both of these people are doctors, right? And so, what is a layperson supposed to think? Path forward, what I generally tell clinicians is rely on the Canadian Immunization Guide, which is amazing. The Ontario Immunization Advisory Committee makes Ontario specific recommendations. Um, but in general, what I end up talking about with a lot of people are, base sort of like baseline it's not baseline's not the right word. Fundamental um, infectious disease things. How does one acquire vaccine preventable infections? It doesn't matter if everybody's immunized, you can still get tetanus from the soil. How common are different vaccine preventable infections in case parents are like, well, I want one, but not two. You know, it's hard. The chance of getting polio in Canada right now are extremely low, whereas the chance of getting pneumococcal infection are a lot higher. And what are the consequences of infection, right? So pertussis is more common than meningococcal disease, but as long as you're not a young infant, you probably won't die of pertussis. Whereas you get invasive meningococcal disease, you have about a 10% chance of dying off the top, and then a quarter of the survivors will have bad things happen to them. So it's tough to say. Most families are worried most about safety. And so something that I come back to constantly is that vaccines are just about the safest interventions that the medical system has ever come up with or will ever come up with, right? And the degree to which they are, scrutinized before and after licensing dwarfs anything else. You know, this is a major contrast to antibiotics, which have numerous obvious, documented, evidence proven adverse effects that are a big deal. Antibiotics are bad. I mean, they're good if you have a severe infection, but like to pick an antibiotic instead of a vaccine in almost all, is a bad idea, right? It's almost like and often you get this vaccine hesitancy um, from people who want to do other medical stuff. Like I was just reading something in the New York Times yesterday about ivermectin, about how it's going, they're making it over the counter at so many places, and people are taking ivermectin every day. Like, I think ivermectin's awesome if you have like strongyloides, right? Like, but but these are people who ostensibly believe in medicine, but don't like it's the Mennonite thing is almost not almost is more intellectually honest, at least, because these are people who decide they don't want any Western medicine at all, which is sort of a more, you know what I mean? Like it's a more defensible point to be in. I would say that many adverse events that we talk about are definitely not caused by vaccines. I think many people know that I'm like the original shades of gray person. Like I'm like, well, I never, almost never give anybody a straight answer for anything. You could do this, you could do this. This is good, values and preferences. Vaccines don't cause autism. They do not. That is the one thing that I can be definitive about, despite the fact that the case is being reopened in the United States. Um, but that you can sort of wipe off. There are definitely APHEs that are caused by vaccines, right? Most are benign. Large local reactions don't do anything, even though they occur and they bother people and they come back. Many do not recur. So hypotonic, hyperresponsive episodes are terrifying to parents, but they almost never come back. And so the benefit of continuing vaccination outweighs the potential harm. And serious APHEs tend to be rare. Guillain-Barré is very, very rare. Cardiac toxicity for older teenage boys with COVID vaccines was a lot more common than Guillain-Barré. And those could be serious, but the incidence was, 1 in 5, 1 in 10,000 back then, and nobody's getting two-dose series anymore. And so that's important to talk about. I mean, what I would do to improve vaccine uptake in patients is do the quote simple things first. If the reason the kid isn't vaccinated is because the parent has to work, which is like a very common phenomenon, like opening up a clinic after hours or figuring out a way to do something on the weekend is way easier than convincing somebody, you know, who has a rational belief in X, Y, or Z. Oh, framing is so critical. So we have this amazing new, RSV monoclonal antibody product, nirsevimab. I have no connection to Sanofi, by the way, no connection. Um, but Spain got more than 90% uptake. I was lucky enough to work in a hospital that dedicated resources to rolling this out. And we had like a world RSV leader who like volunteered to coordinate everything, Dr. Bosco Pays. You know what I mean? There were so many people. It was so great. And yet our uptake was at like in the mid 70s on the neonatal wards. And I was like, what? Why? And the answer was that new parents were often too, fatigued, emotionally overwhelmed to have a discussion about a new vaccine. Which I mean, I've never had a baby come out of me, but that I get. But it was being framed like it wasn't like I think a lot of the issue was framing. Like if you go to some person who's just given birth and say, "Hey, do you want to talk about this new vaccine that we might give to your baby if you want to?" I could totally see people saying, "Well, we can't talk about this." Like, you know what I mean? Well, I'll talk about it with my family doctor. If you either lay the groundwork before the kid's born, or you go in and say, "There's this great product which has amazing effectiveness to prevent against the most serious respiratory infection in pediatrics that predominantly affects young infants," you probably would get a bunch more uptake. I think a lot of it is listening and giving people time to talk, which is sort of easily for me because I'm a subspecialist, harder for primary care clinicians who are paid by the government to spend 8 minutes with people. It's good to review references in case people say things that seem insane. And what I end up saying in a lot of these talks is that you got to try to avoid this sort of us versus them, this whole adversarial thing that comes up, and say, like, I often explicitly say, like, we're all trying to find the strategy, the path that has the most benefit and the least risk for Johnny, right? Like we're all doing this together. And I think that helps to get people on side. That's what I got.
Thank you. I'm going to turn it over to Sam. I just want to mention, because I forgot to, that if you have questions, we'll answer them at the end of the talk. And our Zooms are now set up so that I can unmute you so you can actually ask your question as opposed to having to type it. But I still need to know that you have a question, so you'll have to raise your hand. Okay, Sam, over to you.
All right. Thanks very much. So I'm going to be talking on the science aspect of things and how we here at McMaster have been working on developing sort of the next generation of vaccination strategies for some of the toughest respiratory infectious diseases. So when it comes to developing the next generation of vaccine strategies, there's a lot of things we take into consideration. Things like, the flavor of immune responses that a vaccine generates. Where do we want these responses to be generated by taking into consideration the route that the vaccine's given, safety and reactogenicity of our vectors, and then a handful of non-immunological considerations, manufacturing and scalability, population and epidemiological considerations like hesitancy, and then economical considerations like the cost of these vaccines and upscaling to be able to meet the demand that's required. So when I think about the effectiveness of a vaccine, I don't think that it's just related to its ability to confer protection. I think effectiveness is quite intimately linked with its perception and uptake by the population. So when we talk about individuals who are vaccine hesitant, who, range from being refusers to cautious acceptors, factors like reactogenicity, how that vaccine is delivered, any safety signals that come up as a vaccine is being deployed can cause major shifts in the probability of these individuals either accepting or refusing these new vaccines that come onto the market. One of these factors is the fear of needles and injection pain. So that hesitancy for essentially needle phobia can lead to altered vaccination schedules, delayed acceptance, refusal. And some systematic studies have showed that among children and parents of children, that hesitancy of up to 5 to 13% is attributed to the fear of getting a needle or the pain associated with it. And I think what's really interesting is that during COVID-19, when there was a range of studies carried out in the United Kingdom, greater than 25% of the people who were part of these studies had some level of blood injection injury phobia. And of that 25%, 22% were actually hesitant of getting the vaccine because of the fear of getting a needle. Now, with that being said, our vaccination strategies during COVID had resounding beneficial impacts, right? So the deployment of our first generation vectors kept millions of Canadians out of the hospital, prevented hundreds of thousands of deaths, and had major economic benefits. But there's always room to improve. So when it comes to addressing the gaps that we know exist in not only the vaccines themselves, but how they're administered and the public perception, we really have to build on the foundation of our first generation vectors and improve them to cover all these aspects. I'm going to spend a few minutes talking about the importance of immunization route and some of the considerations when it comes to how we deliver vaccines, right? So we'll have immunological considerations, and just as important, what are the devices we have available to deliver these vaccines via different routes? So work that we've done over the last 2 decades and work done by others has really provided some concrete evidence highlighting the importance of immune route and immune cell geography. So the majority of vaccines in the human immunization program are given systemically, either intradermally or intramuscularly. And we know that this route is able to induce quite robust vaccine-specific adaptive responses within the periphery. But when we sample the airways, whether that be the upper respiratory tract or the lower respiratory tract, there's actually quite a lack of vaccine-specific T cells or B cells in those regions. So intramuscular vaccination largely fails in inducing long-lasting mucosal immunity. In contrast, when a vaccine is given directly into the respiratory mucosa, not only are we able to recapitulate that robust systemic response, but with the vaccine being deposited in the actual respiratory mucosa, we're able to engage bona fide tissue-resident immunity within the airways and within the other parts of the respiratory mucosa. And this is classified by tissue-resident memory B cells and T cells that provide long-lasting memory immunity, and as well as local antibody responses as well. And these mucosal responses we've seen are not only important in preventing transmission, but protecting against severe disease. So that's the immunological aspect. How do we actually deliver a vaccine mucosally, and how do we ensure that we're engaging the proper type of immune response? The short answer to that is when you're delivering anything to the lower respiratory tract, the size of the droplets plays a very critical role. So studies have shown that to have efficient deposition within the lower respiratory tract, and in particular, when we're looking to engage immune responses deep within the lungs, you need to have aerosol droplets that are less than 5 microns in size. That allows deposition in the tracheobronchial regions and alveolar regions. So we need devices to be able to nebulize vaccines to this droplet size. And there's been quite a bit of foundational work done right here at McMaster, particularly using the Aerogen nebulizing devices. So the Aerogen device itself has a vibrating metal mesh that's perforated. And when an electrical current is put into that mesh, it vibrates at a frequency that turns a liquid formulation, like a vaccine, for example, into a fine mist of particles that are 1 to 5 microns in size. That size, once it's inhaled, allows for sufficient deposition within the alveolar and the lower respiratory tract and allows us to engage immunity where we need it to be established. So our institution continues to be one of the leading institutes in developing next generation inhaled aerosol vaccines. Our platform has really over the last 2 decades been founded on adenoviral-based vectors due to their high genetic plasticity, meaning that we can incorporate multiple antigens of interest for the pathogen we're targeting. Adenoviruses have natural tropism to the mucosa, meaning that they're excellent vectors for engaging mucosal responses. And endogenously, ad vectors are actually quite immunogenic, meaning that you don't need to add adjuvants for these vaccines to be potent in engaging immune responses. And I'm going to spend some time talking about, where have we come from in terms of our inhaled aerosol strategy. So our foundation was actually in the tuberculosis field. And when the pandemic hit, we shifted our knowledge and technology to COVID-19. So TB is a bacterial infection caused by Mycobacterium tuberculosis. It primarily affects the lungs, and it's exclusively spread person to person through inhaled aerosol droplets. Infection with active disease is associated with formulation of these granuloma and granulomatous structures within the lungs, which eventually, as they grow and evolve, can not only damage the vasculature, promoting systemic spread of the bacilli, but can destroy conducting airways, facilitating spread. And it's not really talked about here in Canada, but TB remains actually a major global health burden, and it's also a local burden, particularly within our territory. So in areas such as in Nunavut, the rates of tuberculosis for active disease are nearly as high as we see in the TB endemic regions in certain parts of sub-Saharan Africa. So it's a major health issue in Canada, but it's underrepresented, unfortunately. And all of this is to say it's under the realm in the context that we already have a vaccine available for TB, which is now over a century old. So this is the BCG vaccine. And, this vaccine has been incredibly effective against disseminated disease, but it continues to fall short in protecting against pulmonary tuberculosis. And the reason for that, and what I had hinted to before, is that these intramuscular and intradermally delivered vaccines, they fail at inducing airway responses. So in the context of a respiratory infection, such as with MTB, by the time infection occurs and the delay needed to bring these immune cells from the periphery where they're generated by intradermal vaccination into the airways takes too long. And that gives enough time for the bacteria to establish a foothold. So you get improper or a lack of protection with these intramuscularly, intradermally delivered vectors. And the grand challenges that have been always highlighted by the Bill and Melinda Gates Foundation have continued to push not only for improved vaccine strategies for tuberculosis, but also one of their top directives is developing needle-free strategies. So we've done extensive work originally in the lab using a human adenovirus serotype 5 vector expressing what we would consider a prominent MTB antigen. This was antigen 85A. And over 2 decades of research with this vaccine, comparing it either delivered intramuscularly or via the respiratory mucosal route, we've seen through a range of animal models, ranging from rodents like mice and guinea pigs, all the way to non-human primates, that whereas intramuscular vaccination continues to fail in inducing mucosal immunity, even in BCG primed animals, we can restore that gap by delivering these vaccines via the respiratory mucosal route. This 2 decades of research accumulated to the first in-human trial comparing an intramuscular TB vaccine to one delivered by inhaled aerosol. And what was unique about the trial that we had carried out, so this was in BCG vaccinated volunteers, we were able to sample the airways directly via bronchoalveolar lavage to really understand what is the profile immunity in the human airways following an inhaled vaccine. I'm just going to summarize the findings here. So in terms of safety, this is a needle-free strategy. So obviously we have no local site injection pain or reactions. And the adverse events that we see that are associated with vaccination, such as cough, is one of those prominent ones. They're transient and they're all grade 1 in nature. When we assess respiratory function, not only during the acute phase, but throughout the course of the trial, we see no changes in respiratory function, further exemplifying the safety profile of these inhaled vectors. But immunologically, same thing that we've seen with our preclinical models translates into humans, that intramuscular vaccination fails at inducing mucosal responses. Those are the responses you need to protect that portal of infection from MTB. That gap is filled when that same vaccine is given mucosally through an inhaled aerosol. Now, we wanted to continue developing our ad strategies, but when the pandemic hit, we had to shift our gears in developing and applying our knowledge of inhaled vaccines to SARS-CoV-2. But what I want to highlight is that just because, COVID hit didn't mean that cases of TB stopped, or it's just the lens shifted. And in 2022 and 2022 to 2022, for the first time in over a decade, the cases of tuberculosis were increasing and deaths were increasing as well, meaning that we had reversed years of progress in dealing with this disease. So it shows how delicate and fragile the TB ecosystem is when it comes to healthcare solutions and vaccine solutions. With that said, we are looking at revitalizing this platform, and I'll talk about that later after I talk about where we are with COVID. So when it comes to development of vaccines, often people don't think about how long it takes, but the rationalized paradigm when it comes to vaccine development is through a series of iterative refinements and developments. This takes 10 to 15 years of time, and we've seen that with our inhaled TB vaccine as well. The thing is, during a health emergency or a health crisis like SARS-CoV-2, you need rapid design and implementation of vaccines. So we consider this the pandemic paradigm, where these vaccines are generated in a much shorter time point to be able to protect the population and minimize burden on our healthcare systems. So within that period of 1 to 2 years later, we had approval of multiple COVID-19 vaccines. All of these vectors were designed to express that surface antigen of SARS-CoV-2, which was spike. They were formulated for intramuscular delivery, like the ones that we saw with mRNA vaccines, and they induced quite robust peripheral T cell and B cell responses. And we know that in the early phases of the pandemic, these were remarkably successful in protecting against infection and severe disease. The thing is, there's numerous limitations to these first-generation vectors that became apparent as the virus had evolved. As the ancestral variant began to evolve and adapt to become more transmissible and more immune evasive, we saw that the efficacy of these first-generation vectors started to erode. So we tried to counteract that by developing updated first-generation vaccines that had the spike antigen of the most dominant circulating variant. The issue is, this is an exercise in futility. You keep chasing a variant that keeps changing. So by the time something is manufactured, there's already a new variant that's emerged, and you have to redo your formulation. In addition, some studies have shown that this spike-centric approach in vaccine design was actually providing immunological pressures to facilitate vaccine-mediated immune response evasion. Additionally, like I had talked about before, and some of the colleagues in the field have shown, is that mRNA vaccines don't induce mucosal responses. So that's why we also can see the more rapid waning in protection from these injectables. So how do we improve on these vectors? So a review that we had written a few years ago discussed strategies we can employ in developing next generation vectors. And these strategies apply beyond SARS-CoV-2. So in the context of COVID-19, this spike-centric approach, I think, is no good. You need to include additional antigens that are resistant to the mutational landscape that we see with spike. So we want to generate vaccines that include multiple antigens, expand the breadth of the responses, but specifically induce them at that portal of infection within the lungs. So building on our inhaled vaccine platforms, we had taken a new adenovirus, one that's based on a chimpanzee adenovirus serotype 68 platform, and we developed an evidence-directed trivalent vaccine. So we included spike because we know it's critical in neutralizing and antibodies against spike and neutralizing the virus. But we included two internal antigens, nucleocapsid and RNA-dependent RNA polymerase. So the sequences of these two antigens has remained relatively conserved among all of the different SARS-CoV-2 variants that have emerged up to this day, meaning that T cell responses generated against these antigens are able to recognize a range of these variants. Additionally, there's actually quite a high amount of conservation in the sequences that we have selected for these antigens among bat coronaviruses, which provides us a potential, obviously needs to be shown that this vaccine might be able to provide protection against other coronaviruses as well. So all that preclinical studies that we had carried out accumulated into our now completed phase 1 trial assessing the safety and immunogenicity of our vaccine delivered by inhaled aerosol. So I'm going to show a snippet of this data where we further show how infection or mRNA vaccination fails to induce mucosal responses. And we can fill that gap by delivering a vaccine via the mucosal route. So our cohort in this trial, I'm just showing a part of it here, included previously mRNA vaccinated individuals who've had SARS-CoV-2 infection, mild to moderate infection. We sampled the airways by bronchoalveolar lavage and also took peripheral blood samples at baseline. After we delivered the vaccine by inhalation, we once again did a secondary lavage and blood sampling 4 weeks post-vaccination to understand the changes in the immune landscapes within the periphery and within the lungs. So this included assessing T-cell responses in the BAL, antibody responses, and also looking at similar readouts within the peripheral blood. Now, in terms of safety, these mimic exactly what we saw with our Ad5-based platform for tuberculosis. All grade 1 adverse events that were self-resolving, including some solicited events that were systemic and some that were respiratory. And importantly, when we assessed respiratory function, we saw no changes in respiratory function prior to vaccination and at multiple time points post-vaccination, further exemplifying at least the safety of the vector in our cohort. Next, we looked at the T cells in the airways. So the data that's shown here is looking at our baseline T cell responses against 3 antigens, the ones that are included in our vaccine: spike, nucleocapsid, and the RDRP. Now, these individuals were previously mRNA vaccinated, so that includes the spike antigen, and they were also infected previously with SARS-CoV-2. So infection with SARS-CoV-2 should, in theory, induce immune responses against all these antigens as well. What's interesting is that the airways are nearly completely absent of immune responses against the virus and immune responses you would expect vaccination to induce, which means that there's a lack of mucosal immunity in previously infected mRNA vaccinated individuals. But a single dose of the inhaled vaccine induces a significant boosting of immune responses against all of these antigens. And importantly, when we look at the quality of these T cells that are generated by inhaled aerosol vaccination, the majority of them express tissue-resident memory phenotypes. These T cells are critical in providing lifelong, long-lasting immunity within the airways against subsequent infection. And this was all generated by only a single inhaled dose of the vaccine. Additionally, we looked at whether these T cells can cross-react with variants that have become highly evolutionarily divergent from what originally emerged in 2020. And T cell responses presented here on the left show comparability in these responses, meaning that we've seen no impact throughout the evolutionary course of the virus and the immune reactivity of our vaccine. And similarly, when we look at antibody responses in the airways, our vaccine continues to actually boost responses against this variant, despite the vaccine encoding an ancestral spike antigen. Finally, we also looked at peripheral responses. So mRNA vaccination, we know, or any of the vaccines that are given intramuscularly are able to induce spike-specific responses in the periphery. And we see that here at the top left, looking at spike-specific responses. What's interesting is that even when a vaccine is given via the inhaled aerosol route, and similar to what I talked about conceptually earlier, this inhaled vaccine is able to boost peripheral responses as well. So what that means is that inhalation of the vaccine not only induces local immunity, but it boosts peripheral as well. So all of this work has accumulated to us now starting our phase 2 randomized placebo-controlled trial here at McMaster University. And this is currently recruiting, and we're looking to actually test the vaccine at different sites across Canada. And I think a lot of the data that we've generated in our phase 1 trial has provided quite the resounding support for us to continue the clinical evaluation of this vector. The other thing I want to highlight is the timelines that it took for just the people here at McMaster University to carry this work out. So from preclinical testing in 2021 to completion of phase 1 trial and initiation of phase 2 was just under 4 years of time. So it's at a rate that's far greater than that's ever been achieved here institutionally with a new vaccine candidate. The other thing I want to highlight is that our product, our approaches, these innovations have all been Canadian designed, Canadian developed, and Canadian manufactured. right here at McMaster at Fitzhenry, where we generated our GMP grade quality products, to the Alberta Cell Therapy Manufacturing, where we had our products filed for phase 2, back to McMaster, and then distribution to our sister sites at OHRI and CCFV, where we're carrying out our phase 2 trial. And I think we're very proud of the fact that this is Canadian made, Canadian designed, and implemented in Canada. With that, I'd just like to say thanks. All of this work that I'm presenting here is accumulation of the work of dozens and dozens of individuals. And, we really look forward to the next chapter, not only in the COVID-19 field, but revitalizing our TB platform and expanding it to other important respiratory infectious diseases. Thanks.
Thanks, Sam. Thank you both for those great talks. Very exciting and pretty cool. All right, I have a question for Sam. I noticed that the responses to 2 of your 3 antigens were pretty robust. Do you want to talk about your
Yes.
RDRP sorry-looking responses?
Yeah, so the vaccine doesn't encode the full length of RDRP. We did epitope mapping and selected a region of RDRP that shows quite high homology, not only among the bat coronaviruses, but it's specific to a certain MHC haplotype that's loaded. Our N number for phase 1 is low, so there's a probability that the haplotypes of the people in our trial aren't able to efficiently load that RDRP antigen. So we're hoping to see engagement of these responses in our larger phase 2. There's also the possible consideration of immune dominance. Spike and nucleocapsid are quite dominant. RDRP is quite a small antigen we chose, so there's a possibility that we're overwhelming it. Then again, we really don't know if quality and quantity actually matters. Even if you have a small boosting, those T cells might be actually just as protective as ones that target a different antigen that are at a greater quality or quantity.
Okay. There's a question from Mike Surratt. Let me unmute you, Mike. There you go. Ask away.
Oh, sorry about that. Great talks, both of you. Sam, I had a question for Sam. Very exciting. I was curious about 2 things. One, in the phase 1 trial, it didn't seem that the previous infection actually incurred any mucosal immunity, or did I misunderstand that? And is
That's
that
correct.
Yeah? And so I was a little surprised by that. Do you know why? And then the second question is with the inhaled aerosols. I assume you probably get some nasopharyngeal response, but do you get upper airway response in general? And would that actually be important?
Okay, so regarding the impact of infection on mucosal responses, this is a tough one to address because there's a couple of considerations. In literature, severe infection is associated with quite an aberrant immunological response within the airways. We see that in ICU patients. Our participants had mild to moderate infection. Most of them were previously vaccinated. So it's possible that the magnitude of responses generated by infection, the antigenic load of the virus might have been too low to generate a robust response. They could have also waned over time quickly. And the majority of our breakthroughs are with the Omicron subvariants, which primarily impact the upper respiratory tract. So lower respiratory tract responses remain rather limited, unless in cases where the virus can disseminate to the lower respiratory tract, where you have manifestation of severe disease. So those are the couple of possibilities. Nasopharyngeal deposition, we've never looked at. Is upper respiratory tract immunity important? Absolutely. We want to assess that. And if we all believe in the common mucosal immune system, respiratory responses in the lower respiratory tract should mimic what we see in the upper respiratory tract, but we haven't looked at that yet. And it's something that is of interest. In our preclinical models, yes, but preclinically that's delivered intranasally. So the vaccine is exposed to the nasal mucosa. Whereas in this one, we have the nasal cavity. We essentially have a nose clip on, so people can only breathe it in, and you can't get any of the particles up into the upper respiratory tract. But it'd be of interest to know. We just haven't looked at it.
Yeah, just with regards to the lack of mucosal immunity in the mild to moderate, I think a lot of people believe, well, I've had it, so I have really good immune response. I think it's an important message to get out.
Yeah, exactly. And we see so many breakthroughs and continued infections, and in certain cases, those can manifest as severe disease.
Yeah.
So the long-term consequences we are continuing to still understand.
Thanks.
Very cool. I have a question for Des. When I was a kid, back when dinosaurs roamed the earth, we had nurses that came into schools to make sure that people were caught up on their vaccinations. Is that still a thing? Is this a way for us to, catch these kids up that have been locked out because of COVID?
Yeah, that's a good question. I'll answer it if I can ask a question to Sam after this. So, like there is that program of giving meningococcal quadrivalent vaccine and hepatitis B and HPV in schools, and so that is all public health delivered. But in terms of finding people and catching them up, it's still the same system as that it always was, right? And I don't know if you guys read the Hamilton papers, but there was this recent spike spate of people getting these notices because they had put it on hold for so long that nobody was checking. Overall, I think there's a lot of aspects of vaccine delivery in Ontario that are a real problem. We absolutely need a vaccine registry for like 17 reasons. You know, not only would it enable public health, not only would it enable people to know if they and their kids are up to date, but it would help like the doctor at the emergency. It would help public health, and it would facilitate all of this research that in like, for example, that PHO did during the COVID-19 pandemic to assess the effectiveness of vaccines and to demonstrate what APHIs were related. Like it's an absolute no-brainer, and yet we do not have one. It's not like it's totally out of reach, like Manitoba has one, Denmark, like, I mean, there's all these. So
Mhm.
I think a vaccine registry is really the answer. The problem with public health is been asked to do 17 trillion things. Now there's a measles outbreak. So there's not a whole bunch of extra people hanging around who can go administer vaccines in schools to like the 7-year-olds who aren't up to date. Can I ask a Sam
Problem.
question? Oh my God, it's 9.
You
Uh,
may ask Sam a quick question.
Oh, awesome. Amazing. So, and you might have talked about this and I wasn't paying attention. Is there a thought that you would modify the mesh size if you want to preferentially deposit stuff in maybe the upper airways? Like there's a lot of talk about avian flu now, and there's more receptors up here than there are lower down. Are people thinking about doing that?
Yeah, so the droplet size that's generated with the mesh, uh, that facilitates lower respiratory tract deposition, but it doesn't mean it doesn't deposit in the upper respiratory tract. So we get both throughout. It's more of an issue of getting it lower versus getting it higher.
Okay, I have one quick question from Dawn as well, if you'll indulge us for one more minute. She wants to know what the evidence is that the RNA polymerase and non-spike antigens are correlates of protection. They don't find any evidence in their studies that there are correlates of protection. So she's
Yeah,
curious to
so
know.
there's been work done by Shane Crotty's lab that provides pretty good evidence for the role of nucleocapsid responses in protecting against severe disease, especially in the context of waning spike responses. Our evidence for RDRP actually comes from a really strong paper, I think that was published in Nature, that showed that healthcare workers who were continuously exposed to SARS-CoV-2 during the first wave without any vaccination remained completely healthy and uninfected. And BAL assessment on these individuals showed that they actually had high levels of RDRP-specific CD4 T cells that were contributing to abortive infection in these individuals, and they were able to show that that's what was correlated to protection. So we didn't just choose them for the fact that they're internal. They were actually based on some of this work that was quite seminal when the first few studies came out about correlates of protection against SARS-CoV-2.
Hey, great. Jerry Wright just wants to give you 2 thumbs up. He thought these were great presentations. But since we're over time, I will thank you again, and I'll see everybody next month.
Thanks for the opportunity.
Bye.